Yeah.
Recording.
That's so I can send you things to do the, okay.
All right.
There's 57 slides here. That's scary. Okay.
So Sheldon, how long do we have on the time?
About an hour. We have an hour. I
An
can,
hour.
yeah.
Okay.
Talk
So,
fast.
okay. So I will just quickly introduce the technique we use in the lab and some previous data before going into the preliminary data that we got for the adipose tissue. Okay.
Okay.
So this is the lipidomic analysis that we do in the lab. We use a high resolution mass spec, yeah, and we established this method together with Adriano, Marcos, and Isaías. Isaías is the guy that is probably will join us soon, but so we do this analysis. Adriano is a specialist in mass spec analysis, so he does all the lipidomic analysis in our lab. Marcos is a specialist in the data processing, lipidomic data processing. So he identifies all the lipid species in the samples. And Isaías is help us with the statistic analysis. He is also a neuroscientist, so he knows this AOS disease. So he helps us for in the analysis. So this is the workflow, the general workflow that we use for the sample. So we extract the lipids from the tissues. So, and then after we collect the organic fraction, we evaporate that, concentrate and inject into the mass spec. So, and then after we collection of the LCM SMS data, we do the data processing, statistics, and interpretation. So we've been applying this global lipidomic analysis in several analyses. We do lots of analysis in ALS disease, but we also applied this platform in the analysis of other studies. So dietary interventional studies, Zika virus study, this is Marcos did in babies recently that he published, C. elegans cells. So we applied this platform in several samples in several diseases. So for AOS rat model that we have in our facility, we have so the wild type and the AOS transgenic rats. So we collect samples in two stages, which before symptoms, the asymptomatic group, and after the onset of the symptoms, which appears around 120 days. So we have two groups, the asymptomatic group and the symptomatic group. So we collected several tissues, motor cortex, spinal cord, blood plasma, liver, muscle, heart, and also the adipose tissue. And then we submitted this to global lipidomic analysis. And I will show you very quickly the data on in the spinal cord that we, the motor cortex. In motor cortex, we did not find a difference by the disease. Most of the difference that we have observed in the lipidomic composition was related to age. In the spinal cord, lipidomics, we monitored for about 400 lipid species. And in the spinal cord, we could find signature lipids that were associated with ALS disease progression. So we found an increase in cholesterol esters and also ceramides in this ALS symptomatic animals.
So is that a published finding then, or is that a new finding for you? Is that published Is that known?
This is the work we recently published. Adriano, né, published here in 2019. So this is already published result. And we found quantitatively like an 8-fold increase in cholesterol esters that were linked to polyunsaturated fatty acids. And we know that cholesterol esters are neutral lipids that are stored in lipid droplets in the cells. So this increase reflects the increase in lipid droplets in cells in the spinal cord. And another finding was the reduction or the loss of cardiolipin in the spinal cord. We found about 50% decrease in total cardiolipin content. Cardiolipin is a mitochondrial lipid. So the decrease in cardiolipin reflects a problem in mitochondria. Okay, so these both data, cholesterol ester accumulation and cardiolipin loss, is consistent with the accumulation of lipid droplets that have been already reported for other studies using AOS rat model. A group in the Uruguay have shown the increase in lipid droplets in aberrant astrocytes from spinal cord. And also the loss of mitochondria is consistent with several studies showing mitochondrial dysfunction associated with the disease. So this data was published in the scientific report in 2019. So these are the main signature lipids that were found in the spinal cord, increasing ceramides, decreasing cardiolipin, and increasing cholesterol esters. So we believe that the accumulation of cholesterol esters and mitochondrial dysfunction could be potential therapeutic targets in the central nervous system. So now I move to the lipidomic data in other tissues. So I will talk about data that we have not published yet, the analysis of the lipidomic analysis of blood plasma, liver, muscle, heart, and adipose tissue. So in blood plasma, we found 296 lipid species. And the main changes that we observed that were associated with ALS disease progression is highlighted here. So we found an increase in two types of ceramides, exosylceramides and acylceramides, a decrease in phosphatidylcholine and lysophosphatidylcholine, and a decrease in several species of triglycerides. The decrease in triglyceride is consistent with the hypermetabolic phenotype. This is an expected result. On the other hand, the increase in exosylceramides and acylceramide was something new for us. So we tried to find where these species, these ceramide species, are located in the blood plasma. So we fractionated the lipoproteins in the plasma, and we could find that exosylceramides and acylceramides are enriched in the HDL fractions. So like here, you can see that they are enriched in HDL fractions. So the preliminary conclusion for this part is that exosylceramides and acylceramides are signature lipids that are likely associated to ALS disease progression, but there are still, although we could find that these species are located in HDL, we don't really know what are the real source, the exact source of these species and what are the role of these species in the body. So next we try to go further in trying to link the increase in these species with the lipidomic data in other tissues. So we did a mute tissue lipidomic data integration. So we we compared the data obtained in the liver, plasma, muscle, heart, and also here I will show the data that we have obtained in the adipose tissue. So here is an overview of the main changes that we found in lipids in the different tissues, muscle, liver, heart, and blood plasma. So one thing that took our attention was that the increase in exocylceramides was observed also in the muscle. So the same species, exocylceramide species that were increased in the muscle was also increased in the blood plasma. So we believe that this data suggested that circulating exocylceramides are signature lipids of muscle wasting. And in support of this hypothesis, we found this paper in the literature showing an increase in the exocylceramides in the blood plasma of cachectic patients. So, the levels of exocylceramides was strongly correlated with body weight loss in humans and mice. So this is a data in human cachectic patients. So exocylceramide seems to be a signature marker for muscle atrophy and cachexia. So, the conclusion of this part is that increased exocylceramide that we can clearly see here in the AOS symptomatic animals are likely to be markers for muscle atrophy. So another analysis that we did was this correlation analysis and between liver lipids and plasma lipids. So this figure shows that there are several lipids that are correlated between the liver and plasma. For example, here phosphatidylinositol shows very good correlation between liver and plasma. However, this species was not differentiated between disease animals and wild type. But for instance, here acylceramides, we can see a good correlation between liver lipid and plasma lipid, okay? So, and also a difference in the levels of these species in the symptomatic animals. So we can see that in AOS animals, the acylceramides are consistently higher than wild type animals. So there are different species of acylceramide that are consistently different. On the other hand, exocylceramides that we also saw difference in the AOS animals was not correlated with liver. So we believe that acylceramides are probably formed in the liver. And to support this data, we also found data in the literature that have proved the formation of acylceramides in the liver of mice submitted to high-fat diet. So the proposal of this study was that excess fatty acids are stored, esterified to ceramides, forming acylceramides in lipid droplets. So this study did not show acylceramides in blood plasma. However, we believe that these acylceramides in lipid droplets are delivered to the blood plasma. So so why
Does
that's why we
that make it, is it a toxic thing and that's furthering the muscle atrophy, or is it a result of the muscle atrophy then storing? Which came first?
I believe it's a consequence. It's not easy. I think there is no clear answer, but we believe that this is a consequence of lipolysis because this during ALS, the adipose tissue is lipolytic. It's high, there is a high lipolysis, so there is high amount of fatty acids that comes to the liver, and these fatty acids in the liver are converted to acylceramides. If they are toxic or not, we don't know. If this is
If you could reverse it, if you could take one of your ALS rats that become symptomatic and reverse the hypermetabolism, you would expect to see less muscle atrophy and not loss of strength, right?
yeah. In the end, I will discuss more about this, but I think if we can treat hypermetabolism, maybe we can treat the progression of the.
And you have an idea to treat the hypermetabolism, or do you have a molecule to do that yet?
Not yet, but this study here.
So if we come up with a molecule that we believe reverses hypermetabolism, then you could test it in your system and look at that, right?
Yeah.
Okay,
yeah.
so we might have something that we would share, and it would be just easier, I think, for you to make it there than us send you chemical.
Yeah,
But we're working on a retroinverso peptide that looks in our cultures like it does, but we're not sure yet on the pathway, but maybe we don't care because maybe you could figure that out for us.
But see, Shoban, what is very interesting and important, I think, in the data Sayuri is presenting is that we have some very, very classical markers of the disease. So anything that, reverse the condition of hypermetabolism for us will be easy to, check and figure out whether it's working or not.
So what we're looking at is mitochondria and ATP, extracellular ATP, and we can reverse the extracellular ATP back to a normal state. Would you not expect that same thing to work in your hypermetabolic mouse then?
Perfect.
Yeah.
Okay,
correct.
so let me get the okay with Alan Wrights and that we could share with you the sequence, and you just get with I think we used GenBio or GenScript to make it. So it's just it's a peptide. The peptide is stable orally for 7 days, so you can just give it to them in the water. It's we have the it's not toxic, and it's I think 5% absorbed, and we're just taking it now into a Jackson mouse model to see if we change the disease in the G93A mouse, so it will be similar to what you're doing. So let me get the okay with him to share the sequence, and maybe you could do that and add that to some of your experiments, because I understand this is all data from dead rats, so now you're moving into live rats, so maybe you could add another cohort that would be then treated, and okay.
Seems to be a very interesting idea. So to summarize this part, so acylceramide is produced in the liver and delivered into the circulation. It seems to be a marker for excessive lipolysis in the adipose tissue. And exacylceramides are both increased in muscle and in the circulation. So it seems to be a marker for muscle atrophy. So now I will show you the data on the adipose tissue that we promised to you in the project. So we collected subcutaneous fat, the inguinal fat, and two visceral fat, epididymal and retroperitoneal fat. This tissue was collected in 2015. It's quite old tissue. So I showed already to you this data, but adipose tissue is drastically reduced in ALS symptomatic animals. We can see here about 50% loss in adipose tissue, in all adipose tissue in these animals. And the lipolysis rate in isolated adipocyte was higher in the symptomatic stage in the epididymal and retroperitoneal tissue, in inguinal also, but differently. And here is the data on the lipidomic analysis of this adipose tissue that was stored at -20. And so there was a very little changes in lipidome in the epididymal fat, just a high free fatty acids. The inguinal adipose tissue, we saw low triglyceride levels that is consistent with the lipolysis, increased the lipolysis rate and high free fatty acid. And in the retroperitoneal fat, we saw differences in the several free fatty acids, and also oxidized fatty acids that we could find either free or esterified to triglycerides. We found that this result in the retroperitoneal fat was quite surprising for us because it's not so usual to see oxidized fat in vivo, I mean, in these tissues. We don't know if these oxidized fats were formed due to this storage period, or this tissue has less antioxidant defense compared to other tissues, so they are more easily oxidized. We don't really know the source of this oxidation. But anyway, linoleic acid, we found oxidation in linoleic acid, and oxidized linoleic acid could be marker of oxidative stress, could be also a result of chronic inflammation. And we know that there are some oxidized linoleic acid species that act as a signaling and hormonal species. So we, based on this data, we thought that it would be important to check whether we can find oxidized lipids also in the circulation. So we did an oxylipidomic analysis, a method that we can detect up to 200 oxidized lipid species. And for our good surprise, we indeed found a difference in oxidized fatty acid in ALS blood plasma. So the main changes, we could find two main groups of oxidized fatty acids. One that we call as lipokines. It's a hormonal, it's a fatty acid, oxidized fatty acids that are synthesized in the adipose tissue and has hormonal effects. And they were altered. They were increased in the asymptomatic stage and dramatically decreased in the symptomatic stage. So, and also increased levels of several oxylipins and oxylipins that are known to be markers for oxidative stress and inflammation. So these data show us that, as expected, there is in AOS rats, there is an increase in oxidative stress and inflammation. But what called our attention was the alterations in these lipokine levels. So this is the summary. I will come to this.
The blood plasma then was in vivo. It wasn't the stored samples, right? You went back to a live mouse for that, or live
No,
rat?
these plasma samples are storage samples, but
Okay,
are the more recently collected ones.
okay.
This adipose tissue was quite old ones, but the blood plasma are a stored one, but stored at minus 80 degrees and all with all this
Is
care to not to oxidize and so on.
Okay, and is the lipokine, since it's in the circulation, can that cross the blood-brain barrier? And can that act like an insulin type of drug?
That's a very good question.
Very good
There is very little information in regard to the connection of the circulation and the blood-brain barrier for this kind of fatty acid. It is produced, though, it is known to be produced by brown adipocytes. So, for example, if you put them in a cold, the brown adipocyte is going to be like producing this type of lipokine that gets into the circulation. Whether it has a pleiotropic effect that goes into the central nervous system, we don't know. That's basically the answer.
And that's something then you're going to try and investigate. So you would go into so that's the next, okay.
Yeah, this is something we wanted to investigate, this if the lipokine alterations in ALS rats and their role. I think it's something we found that is very interesting because, as Marco said, there is a recent report that showing that these lipokines are released by during cold. If mice is exposed to cold temperature, there is a signal in the hypothalamus that leads to an increase of these lipokines. And like these lipokines are thermogenic, they enhance the uptake of fatty acids from the blood, and then this is important for the thermogenic ability of the brown adipose tissue.
So to see if this is important in ALS patients, you don't need fat tissue then. You've already made the correlation from fat to serum, so you just need plasma from ALS patients to then see, and then we could go after whether it was a cytokine that had something to do with insulin and inflammation then in the brain. It would be an inflammatory response in the brain. Is that right? So we're just getting this our big project going, and I had to cancel that meeting, but we're going to have it at the end of the month as soon as we get a couple more things in place. But you'll have access to 192 well-described patients. So I'm not sure that frozen tissue, even spinal cord or brain, helps you in this. You need plasma, correct?
Yes.
Yeah. Plasma will be very interesting, a good place to see all these changes. Yeah, because
Okay.
Sayuri, can I interrupt just a little bit?
Yeah.
Sure, but I think, working on patient is very hard because, in ALS, you have all sorts of patients, right? Patients that aren't right in the beginning, patients that are super symptomatic, and so if there is among this 192 patients a progression of the disease very clear, established, then this plasma analysis will be like it will work like a glove to us. I think it'll be very
we're to the point where they're getting the funding to get the people in place to categorize and catalog everything, and we'll be taking it to almost every kind of analysis we can possibly do. But yes, there would be serial samples on some of these patients from beginning to post-mortem. So there's probably going to be quite a bit that might be useful to you. And They even have some cells on some of these patients that are immortalized and you know, like ISPCs, and those will be available to everybody. So when we have that discussion, you can kind of check off what might be useful, and then you could circle back with Tipu. Tipu Sadiq's going to be managing it, and so you can circle back with him on what might be useful, and he listened to what you were doing and where you were. He's going to make these samples available to people that have shown some progress, not just anybody that wants to do something, but if somebody's shown some progress with a logical hypothesis and does good work, then that's what he would do.
Right.
Okay.
Very interesting. So to conclude, just to show some more hypotheses on this issue that we were discussing. So we believe that treating hypermetabolism could be a good therapy for survival, increasing survival of ALS patients. And this is already related to these several studies showing that adipose tissue is the level and distribution predict survivals of ALS patients. And also I highlighted here that that despite weight loss, ALS patients shows symptoms, shows hepatic steatosis, and this seems to be correlated with our data showing increase in acylceramides that might be a marker for increased steatosis, lipolysis in adipose tissue. And also here glucose intolerance, the exocylceramides that we saw in the plasma of ALS animals are known to be capable to induce glucose intolerance and insulin resistance. This was a study that was done in mice, but this study shows that increased amounts of exocylceramides impair insulin receptor function, because this leads to increasing gangliosides, and there is a detailed mechanism in this paper. But anyway, the main effect is this insulin resistance phenotype. So this is the pathway. I think we don't need to discuss right now. And these are the lipokines that we mentioned to you that are produced from linoleic acid. This is the pathway, and the lipokines are these two compounds that has these two hydroxyl groups, and they have been shown to be activated in cold and have thermogenic activities through increased uptake of fatty acid by the CD36 receptor. And this is the data that showed this effect, this thermogenic effect of this lipokine. So yeah, and this is the summary and the next steps, what we should do next. I was just not sure if we needed to go further in the adipose tissue, because we promised this in our project that we write to you, but we don't really know if this is really something interesting.
If it's not necessary to confirm it, then I think you move to the next step that would be useful, that you've already done that point, unless you felt that what you've done is not, if it stands up to peer review, if you felt you could publish it and stood up to peer review, I think you can move on. But if you're tenuous about what your results are, maybe if you just did one experiment and took some of your frozen tissue and showed that was equivalent in 1 or 2 mice to fresh tissue, that would be significant to stand by what you found.
Yeah. So anyway, I think the idea to look at these markers in the samples that you mentioned, it will be very, very much productive then. So yeah.
And how about our chemical then to try?
Yeah. This is something that we can do in this rat model and see if they modulate these markers. I think this is really a great idea also. Yeah.
Okay, so I will loop you communicating with Alan Wright. So, I mean, I asked him to do something specific for me, which he did do, and then we gave that chemical to Lauren Kokai, and then she's looking at mitochondria and effects on mitochondria in an ALS patient's adipocytes, and the ASCs that she's got are hypermetabolic, and the chemical we're using, we use 3 or 4 different things, but the retroinversion peptide that we wanted to make was the best one. So now we're moving that into a study at Jackson to say, do we alter the phenotype or extend life? They've got the drug, they've got the mice, they've got the money, so I would imagine they're going to start soon, but that's 3 months away. It doesn't mean that you couldn't try it there. And we're guessing on dose. I mean, I don't know how you decide with a new chemical and an effect. So I think we're going to use 3 and 10 mg per kg. You can go to 40 mg per kg. So we couldn't tell you a dose, but you could start with what you thought. So let me get with Alan to talk to you and see, and just give you the chemical. I think it was about $3,000 for me to make 5 grams, so it wasn't that bad.
Peptide, I could not really understand. Is that peptide?
It's a peptide, 5 amino acids. So it's the active site of thymopoietin, and we've made a retroinversion of it as D amino acids because we wanted to extend the binding. So we did get 120 minutes in serum from orally dosed, where if you look at thymopoietin itself, you have a 40-second half-life in people. So extending it. So now in vitro, it reverses the hypermetabolic state in cells, but who knows what it'll do in an animal. And it's not toxic. We know it's not toxic. And we can give you the data that we have showing how, well, we compared 3. We compared it to a L-amino acid. We compared it to an L-amino acid with reversed, and then we did the retroinversion with the D peptides, and that is the best. We're also looking at it in vitro in PBMCs to see what cytokines can we up and down regulate and if it's doing anything there. We pretty much think it does not have any phosphokinase activity. That's about as far as we've gotten with that.
And is it given in the water or food?
Water. We're going to put it in water, in acidified water, and you can mix it up. And I think we were going with an every 7-day change in the water bottles every 7 days, but I think that they might be shortening that, and I don't remember why. Some things just go over my head, so I have to think about why they wanted to shorten when they were changing the bottles of water. But let me get Alan talking to you and see if that's something that we could do. But I think if you want to use the money we gave you to move on, I'm all for that, and not repeating something you're pretty confident in.
We could use this money to do this. I think this study with the peptide. I think it's quite similar in terms of animals that we were supposed to use. So I think.
Well, only if it's useful. Now, in the mouse, in Jackson's study, we're doing the traditional things. We're not looking at any lipid tissue. They're not collecting any lipid or anything. I don't think they're not too thrilled about trying to get adipose from these skinny mice at the end. So that's why it isn't, that's why it wouldn't be repetitive of what Jackson's doing. It would be totally different. So I think it's worth the money to do it if it's not repetitive.
And is this peptide something published? It's patented?
No, Alan's going to. I think Alan will patent and publish it, but we don't have any data yet. But I feel like Neurodegenerative Disease Inc. wants to move things through faster than worrying about people's IP and that kind of thing. So if we have something that may answer a question, especially if it might get to the insulin signaling part, it would be very useful.
I agree. I totally agree.
Yeah, but I don't see that people can't collaborate and everybody can be on everybody else's paper and do all kinds of stuff. But I'm not willing to say, gee, Alan, wait a year or two till we do stuff before we give it to you. So we would just give you the sequence and the source, and so you could get it made the same way we did with the same. I think we're going with a hydrochloride salt is what it is. So
I think we should discuss these details afterwards. But yeah, very interesting.
okay.
Shilpa,
Yes.
I hope you know the main message here was to show you what we're capable of in terms of analytical analysis of fat.